Let's continue our discussion of antigens. We'll look at the two major types of antigens, and then we'll investigate how the host responds to these two different types of antigens. We'll also look at what antibodies can do. Antibodies have several different functions, and we'll take a look at some of those. And then we'll make some notes of important things that you should be aware of concerning the immune response to certain types of antigens. You'll remember that an antigen is any foreign molecule that's recognized by white blood cells. So when you think of antigens, you should think of any microbe, essentially. All of the molecules that make up the structural components that aid in metabolism, that are involved in cell signaling, all of those molecules are types of antigens. Viruses also have antigens. The molecules that adorn the surface of this HIV particle are an example of an antigen. There are two basic classes, two broad classes of antigens. And the way that the host responds to these two types of antigens is different. So first, let's start by defining these two different types of antigens. To understand this, we're going to look at this cartoon schematic of a bacterial cell here represented in pink and some of our own cells represented in light pink. On the previous slide, we looked at some of the different types of molecules that can act as antigens. And one thing that we noted was that a bacterial cell can make all kinds of molecules. So bacteria produce proteins in their ribosomes that are floating around in the cytoplasm. These little symbols here represent different proteins. Some of these proteins will remain intracellular, but others of these proteins will be exported to the surface. All of these molecules, these surface molecules and these intracellular molecules, are an example of an exogenous antigen. The reason why they're referred to as exogenous is because they are generated, exo is short for outside, they're generated outside of our own cells. So an exogenous antigen describes any molecule made by a foreign cell that is capable of stimulating our immune system. Sometimes our own cells might produce antigens. And when these molecules get produced, they're exported to the surface of the cell. These are examples of endogenous antigens. Endogenous antigens are antigens that are generated inside our cells. So they're molecules made by our own cells that can stimulate the immune system. You may be wondering why our cells would produce wonky or foreign molecules. You'll remember that when we learned about viruses, viruses can gain entry into our cells, and once inside, they hijack the host's replicative machinery and basically force that cell to start producing viral proteins. These viral proteins then later assemble into viral particles. And these are examples of endogenous antigens. Another example are tumor proteins. Tumor proteins occur when there's something awry in the cell cycle. And maybe the cell cycle starts running really fast and starts producing these wonky proteins, these abnormal proteins. Those tumor proteins are another example of an endogenous antigen. The way the host's immune response deals with an exogenous antigen is very different than the way that it deals with an endogenous antigen. So we're going to look at the differences between how the immune system handles these two different types of antigens. And we're going to start by looking at how the immune system responds to exogenous antigens. One of the first cells that a bacterium might encounter when it enters our body is a dendritic cell. Dendritic cells are the lookout cells of our immune response. They are the sentinel. They're found in linings of the mucous membranes, all the entry points in the body, the skin and the lungs. And they're kind of patrolling through, sampling the environment, and just making sure that everything's okay. Dendritic cells are outfitted with a receptor called a toll-like receptor. And we learned about toll-like receptors when we talked about the innate immune response. So toll-like receptors help dendritic cells to recognize foreign molecules. In addition to the toll-like receptor, dendritic cells also have an MHC2 molecule. MHC2 is a very significant molecule because it's found only in antigen-presenting cells. There are only 3 types of antigen-presenting cells: macrophages, B cells, and dendritic cells. So only macrophages, B cells, and dendritic cells contain MHC2. So let's recap. Dendritic cells are the lookout cells of our immune response. For those of you who are history buffs, you could think of the dendritic cell as the Paul Revere of the immune response. You remember Paul Revere was the man who rode his horse in the night to warn the revolutionaries that the British were coming. Dendritic cells perform a similar function. They warn the T cells that there might be bacteria in the area. Dendritic cells are found in the skin, the nose, the lungs, and the intestines, and basically any entry point in the body. And it's for this reason that these cells are likely to be the cell that bacteria first encounter when they make their way into our body. Dendritic cells help T cells to decide whether or not they should become activated. And what that means is that they might present normal molecules to T cells or abnormal molecules to T cells. So to understand how T cells can be activated by a dendritic cell, we're going to take a look at 2 different scenarios. One involves a molecule that's okay. This is probably a self molecule. And the other involves a molecule that's bad. And this bad molecule is an example of an exogenous antigen. So first, let's begin by looking at what happens when a dendritic cell encounters an okay molecule. The first thing that happens is that dendritic cell takes that okay molecule inside. So it phagocytoses that okay molecule. And this is something that dendritic cells are continuously doing. They're sampling their environment. And oftentimes, they will take in self molecules. Once inside the cell, the OK molecule gets chopped up into a bunch of little tiny fragments. And these fragments are just the right size to be loaded up onto an MHC2 receptor and displayed on the surface of the cell. So to recap, the antigen-presenting cell, in our example, the dendritic cell, takes in a molecule. That molecule gets chopped up into a bunch of tiny fragments and loaded onto an MHC2 receptor and presented to helper T cells. Helper T cells in the area will come by and line their T cell receptor up with that molecule in the MHC2 receptor. Remember that helper T cells are outfitted with CD4, and it's the job of CD4 to stabilize the interaction between the TCR and the MHC2 molecule. If this is an OK molecule, the helper T cell will not bind. And therefore, the helper T cell is not activated. So the helper T cell is not activated if the dendritic cell is showing an OK molecule. Now let's take a look at a scenario in which the helper T cell might become activated. And this involves our exogenous antigen here. One way that dendritic cells can detect exogenous antigens in the environment is by their toll-like receptor. You'll remember that toll-like receptors can recognize generalized repetitive patterns on certain antigens. When a toll-like receptor recognizes an antigen, something very important happens. It causes that dendritic cell to express a molecule called a co-stimulatory molecule. That co-stimulatory molecule gets exported to the surface of the dendritic cell. The reason why the co-stimulatory molecule is so important is because it serves as a danger signal. So we probably should depict this as a bright fluorescent orange receptor, because it's indicating that there may be something awry. In addition, when that toll-like receptor is bound, it induces that dendritic cell to phagocytose that antigen, chop it up into a bunch of little tiny fragments, and like before, load one of those fragments up onto MHC2 and display it on the surface of the cell. The major difference between this scenario and this scenario, however, is that now we have a co-stimulatory molecule also next to this MHC2. When a helper T cell is patrolling by, two binding events might happen here. The first is binding of the receptor for the co-stimulatory molecule, which is found on the surface of the helper T cell, to that co-stimulatory molecule on the surface of the antigen-presenting cell. And the second thing that can happen is that if that T cell receptor recognizes that foreign molecule, then it will bind. And those two events cause the helper T cell to become activated. So to recap, helper T cell activation occurs only if the antigen-presenting cell is showing it both a foreign antigen and a co-stimulatory molecule. And this co-stimulatory molecule is expressed by the antigen-presenting cell when its toll-like receptor is engaged. After that helper T cell has been activated, it undergoes clonal expansion. And what clonal expansion is when that activated cell starts to make copies of itself. These copies are a type of immune cell called an effector cell. So this is a helper T cell effector cell. Effector cells are a type of immune cell that are poised and ready to carry out their function. And it's the function of helper T cells to coordinate all the other cells of the immune response. So you can think of helper T cells as the maestro of the symphony. Whenever there's an infection, there are lots of different types of white blood cells that are infiltrated to that site. And among these are B cells. You'll remember that B cells are also a type of antigen-presenting cell, meaning that they too possess an MHC2 molecule. B cells are capable of binding to soluble antigen, and that means that they can bind directly to free-floating antigen that might be in the environment. The way that B cells recognize foreign antigens is by their B cell receptor. The tips of the B cell receptor recognize a very specific epitope on that antigen and bind. This binding event then causes that B cell to phagocytose that antigen, chop it into a bunch of little tiny pieces, and load one of these fragments onto MHC2, where it will be displayed on the surface of the cell next to the CD40 molecule. If this helper T cell has a TCR that recognizes this epitope, then it will bind. When that T cell receptor binds, it causes that helper T cell to do two things. The first is that it expresses the CD40 ligand, and the second is that it releases cytokines. Those cytokines bind to the cytokine receptor on the surface of the B cell, and those two events cause that B cell to become activated. Don't worry, we're going to recap all of this now. B cells are capable of recognizing free-floating antigen. So if there's an exogenous antigen in the environment, that B cell receptor will bind to that antigen, take it in, and chop it up into a bunch of little fragments. Those fragments are presented on an MHC2 molecule to a helper T cell. If the helper T cell agrees that that antigen is foreign, then it'll activate that B cell by doing two things. The first is that it produces a CD40 ligand, or CD40L. A ligand is just any molecule that binds to another. So in this case, we know that the helper T cell is producing a molecule that will bind to CD40 on the surface of the B cell. And in doing so, it's communicating with that B cell. The second way that helper T cell communicates with that B cell is by releasing cytokines. You'll remember that cytokines are a chemical message that gets conveyed to other cells, and it tells them what to do. So these cytokine receptors on the surface of that B cell receive that chemical signal and allows that B cell to hear what the helper T cell is telling it. Those two events are responsible for activating that B cell. After that B cell has been activated, it undergoes clonal expansion. So it produces lots of copies of itself. And these cells will differentiate into two different populations. The first is a type of effector B cell called a plasma cell. So plasma cells are a type of effector B cell, and they produce antibodies. The second type are memory B cells. Memory B cells are important because they can remain in the blood and in the lymph even after the infection is over. If they encounter that antigen a second time, they won't need confirmation from the helper T cell to do something about it. So they're very powerful, very potent cells. Instead of waiting around for helper T cell confirmation to begin production of plasma cells, these memory B cells immediately and very quickly differentiate into strong plasma cells after they encounter an antigen. The reason why memory B cells can so swiftly differentiate into antibody-producing factories is because they've essentially fine-tuned their hearing by increasing the number of cytokine receptors that are found on the surface of the B cell. This allows them to be more responsive and also more sensitive to the cytokine cascades that are being released by the cells that initially encounter a pathogen. Upon receiving these chemical messages, they're not only able to differentiate into plasma cells more quickly, but they can also more effectively home to the site of infection. So all of these characteristics are what makes these memory B cells such potent killing machines. Plasma cells are a type of effector B cell that are responsible for producing antibodies. These antibodies are the bullets of the immune response. So these are the things that are going to result in bacterial death. And there are lots of different ways that antibodies can kill bacteria. When a plasma cell produces antibodies, each of these antibodies will recognize that same exact epitope that the original B cell bound to. And it will bind to that epitope on the surface of that invading bacterial cell. Let's take a closer look now at how antibodies help to control infection. So imagine now these are the host cells, and this is the bacterial cell. One of the things that antibodies can do is they can bind to a particular molecule on the surface of that bacterial cell and interfere with its function. So let's imagine now that these antibodies are bound to a particular molecule that that bacterial cell requires to adhere to the host cell. With these antibodies obstructing that interaction, those bacteria would no longer be able to bind to our cells. This is an example of neutralization. Neutralization describes when an antibody binds to a molecule and interferes with its function. Your textbook talks about how antibodies can neutralize toxins, and they do so by binding to those toxins and interfering with the toxin's ability to have an effect on our host cells. Another thing that antibodies can do, we learned a little bit about this in innate immunity. Antibodies can bind to complement proteins and trigger the complement cascade. Remember, this is the classical pathway of initiation of complement. So the second thing is complement activation. Antibodies are also capable of being bound by a type of cell called a natural killer cell. Natural killer cells have a receptor on their surface called an Fc receptor. And Fc receptors are actually found on the surface of lots of different types of white blood cells. What the Fc receptor does is it's capable of binding to the constant region of that antibody. So it allows these cells, these white blood cells, to recognize an antibody-coated bacterial cell. So it allows the white blood cell to bind and respond to the antibodies that have bound to the antigen. When this Fc receptor on the natural killer cell binds, it causes that natural killer cell to release this nasty cocktail of enzymes, and that causes that cell to lyse open. There's a long fancy name for this entire process involving natural killer cells. It's called antibody-dependent, cell-mediated cytotoxicity, or ADCC. And if you break this big long name down, it makes sense. This is an antibody-dependent process, so antibodies must be present. It's cell-mediated, referring to this natural killer cell. And it's toxic to this bacterial cell. So this is a cytotoxic response. The fourth thing that can happen when an antibody binds to a bacterial cell is that sometimes one branch of this antibody will bind to its respective epitope on one bacterial cell, and this other antigen binding site will bind to another bacterial cell. This causes those bacterial cells to clump together. And this is really advantageous because if these bacterial cells are all clumped together, it makes it a lot easier for a macrophage to come along and just gobble up that entire clump. This process is called agglutination. So agglutination just describes the clumping of bacteria by antibodies. The last way that we're going to talk about that antibodies can help to control an infection is actually something that can magnify to notify almost all the white blood cells in the body. And this way involves a macrophage. Macrophages are one of those white blood cells that possess Fc receptors on their surface. And you'll remember we learned that macrophages also have toll-like receptors on their surface. So this macrophage can bind to this bacterial cell in two ways. Its Fc receptor can bind to the constant region of that antibody, and the toll-like receptor can bind to generalized patterns found in certain antigens. We just learned that when that toll-like receptor is engaged, it causes the cell to produce a co-stimulatory molecule. The recognition of this antibody-coated bacterial cell by this macrophage induces this macrophage to phagocytose the bacterial cell and chop it up into a bunch of little tiny pieces. This process is an example of opsonization. We learned that sometimes when a bacterial cell is coated by a particular molecule, that it can induce other cells to phagocytose it. You'll remember we've already learned about one other opsonin, and that is complement protein C3b. When bacterial cells are coated in C3b, it induces macrophages to gobble them up. It's the same type of thing with antibodies. Antibodies can also act as an opsonin. And when a bacterial cell is coated by antibodies, it increases its chance of being phagocytosed. But this is only the beginning. After this bacterial cell has been chopped up into a bunch of little tiny pieces, one of these fragments gets loaded up onto MHC2 and presented on the surface of the cell. So macrophages are showing this off to helper T cells. If this helper T cell has a T cell receptor that recognizes this antigen and also binds its receptor to the co-stimulatory molecule, it'll cause that helper T cell to become activated. These effector helper T cells have a very far-reaching effect. It's able to communicate with a lot of different types of cells. The first type of cell is a B cell. So helper T cells can release cytokines that cause nearby B cells to become activated. Helper T cells can also activate other macrophages. And when those other macrophages become activated, it induces them to produce nitric oxide. So effector helper T cells can activate macrophages so that they can produce nitric oxide, which is toxic to bacteria. So these helper T cells are putting that macrophage into kill mode. The third type of cell these helper T cells can activate are natural killer cells. And the fourth type are cytotoxic T cells. So you can see that by being opsonized with antibodies, it can trigger a response that has very far-reaching effects. These effector helper T cells can be classified into two broad categories. So helper T cells that tend to activate natural killer cells and cytotoxic T cells are referred to as Th1. Helper T cells that tend to activate B cells then are referred to as Th2. There are a few other details that you should note about how the immune system responds to exogenous antigens. Sometimes B cells don't require confirmation from helper T cells to become activated. We learned that B cells, after they take in an antigen, will load a fragment up onto MHC2 and wait for confirmation from helper effector T cells. If that helper effector T cell then recognizes that antigen, it will express the CD40 ligand and cytokines that activate that B cell. It turns out that some B cells don't require this process to happen. Sometimes in an infection, there might be the presence of really large, bulky molecules. And these molecules are capable of binding to multiple B cell receptors and toll-like receptors all at the same time. And when a certain threshold of BCRs and toll-like receptors are engaged on the surface of a single B cell, it can cause that B cell to become activated, independent of any response from a helper T cell. So that B cell can be activated all on its own. There are names for these two different types of exogenous antigens. An antigen that cannot activate a B cell all by itself is referred to as a T-dependent antigen. And the reason why it's called T-dependent is because it requires a helper T cell to activate that B cell. So a T-dependent antigen activates the B cell only if there are activated helper T cells around. And these antigens are typically comprised of protein. These antigens, on the other hand, are called T-independent antigens because they can activate B cells without any help from a helper T cell. They tend to be relatively big and are capable of binding to several B cell receptors and toll-like receptors simultaneously. Molecules that are big and bulky like this, that have these types of repetitive sequences, tend to be made of lipids and carbohydrates. T-independent antigens generally result in a very weak immune response. And the reason for this is because they're very poor at producing memory B cells. And that's because there is no involvement from helper T cells. Remember that helper T cells produce the cytokine signal that these B cells need to differentiate into memory B cells. Another thing you should note about the way that the immune system responds to exogenous antigens is that antigens that are found intracellularly are still capable of stimulating an immune response. You'll remember that when a bacterial cell is opsonized or coated with antibodies, that this makes that bacterial cell more likely to be phagocytosed by a macrophage. Once inside the macrophage, that bacterial cell gets chopped up into a bunch of little tiny pieces. Now note something here. In this bacterial cell, the red areas represent extracellular molecules, and the pink area represents intracellular molecules. So when it gets chopped up, There are a bunch of little extracellular bits and some intracellular bits too. Sometimes what can happen is MHC2 will load up an intracellular piece and display it on the surface to helper T cells. So in this way, these intracellular antigens are capable of activating helper T cells. So the reason why this can happen is because bacteria are digested, in this example by a macrophage, before they're presented to a helper T cell. Now that we've investigated how the immune system deals with exogenous antigens, let's take a look at how the immune system responds to endogenous antigens. And before we dive into all the nitty-gritty and all the cells involved, let's just get the basic idea. One example of an endogenous antigen is as a result of a viral infection. When viruses make their way into our body, some of these viruses will be neutralized by antibodies. But there will be a few particles that still are able to make their way into our cells and hijack our host cells' replicative machinery to produce more viral particles. It's the job of cytotoxic T cells to recognize these virally infected cells and to release enzymes that kill these cells. So the basic idea is that cytotoxic T cells recognize and kill abnormal cells, like virally infected cells or tumor cells. What we want to do now is learn about how cytotoxic T cells are able to recognize cells that are abnormal. You'll remember that every nucleated cell in our body produces MHC1 molecules. So they're made by all nucleated cells in the body. And what MHC1 molecules do is they take samples of the proteins that are being made inside that cell, and they present them on the outside of that cell so that cytotoxic T cells can come by and take a look. So let's imagine now that we have a perfectly healthy cell producing normal proteins. That cell will take one of these fragments of these normal proteins and display it complexed with MHC1 on the surface. A cytotoxic T cell will come along and investigate this molecule. And if this is a self molecule, it will not bind. Let's imagine now, on the other hand, that we have an abnormal cell. And in this example, the cell is infected with a virus that has hijacked the host's replicative machinery and started making viral proteins. A fragment of these viral proteins gets loaded up onto MHC1 and presented on the surface of that cell. This time, when the cytotoxic T cell comes along, and its T cell receptor recognizes this antigen as foreign, it will bind. Meanwhile, this cytotoxic T cell will be waiting from a response from helper T cells before it takes any effect. And you'll remember one of the things that an effector helper T cell can do is to send a cytokine signal that activates cytotoxic T cells. When this cytotoxic T cell becomes activated, it undergoes clonal expansion. Some of those cytotoxic T cells will differentiate into memory cells, and others will become effector cells. The memory cells will stay in circulation, and if they encounter another MHC1 complex with that antigen, they'll take effect. And again, you'll notice that these memory cells have more cytokine receptors on their surface. And this is what allows them to be so responsive. Now let's take a look at what this effector cytotoxic T cell does. The effector cytotoxic T cells that get produced have a TCR that recognizes that same epitope. You'll remember that this T cell receptor binding to that epitope is stabilized by a molecule of CD8, because we're talking about cytotoxic T cells. So this is a CD8 molecule. This binding causes that cytotoxic effector T cell to release a nasty cocktail of molecules called perforins and proteases. What a perforin does is it perforates the surface of that cell, meaning that it puts little holes in the surface of that cell. And it's through these little holes that proteases are able to gain entry inside that cell. These proteases then induce that cell to undergo apoptosis. So the cell basically dies from the inside. And this is a very neat way of dying. This is a very packaged cell death. So the cell shrivels up and can then be ingested by a macrophage and digested. So effector cytotoxic T cells cause apoptosis of abnormal cells, and those abnormal shriveled up cells then become ingested by macrophages. The reason why we want to have such a neat packaged death to these cells is because if the immune system just lysed these cells open and blew them apart, then that would, in essence, spread the infection. So in this way, we contain these viral particles that may still be inside the cell. Let's take a look at what this really looks like. So here on the left, you're looking at a tumor cell. So you can see that this cell is really large. It's grossly large and very abnormal. It's got all these weird protrusions on its surface. This little guy right here is a cytotoxic T cell. And this cytotoxic T cell's TCR is already engaged, and it's released perforins and proteases. The perforins have put little holes in the surface, and the proteases are making their way into the cell and causing the cell to die from the inside out. So you can see this cell is completely collapsed, and all you're looking at here is the cytoskeleton. Pretty neat, huh? It turns out that some viruses can avoid being killed by cytotoxic T cells. Some viruses are capable of forcing the host cell to downregulate MHC1. And that means that that host cell can no longer express this molecule. In essence, it's a way of binding and gagging this host cell so it can no longer cry for help. With no MHC1 on its surface, these viral proteins that then become produced have no way of being loaded up and displayed to cytotoxic T cells. So cytotoxic T cells will not recognize these virally encoded cells. So they do this by downregulating expression of MHC1 in the host. An example of a virus that does this is HIV. Fear not. There is a backup system, and that comes in the form of natural killer cells. It's the job of natural killer cells to recognize host cells that are not expressing MHC1. And when they see cells like this, they release enzymes that cause that cell to die. What is it then that keeps natural killer cells from killing our normal cells? Natural killer cells are on unless they're turned off. And the thing that turns them off is the presence of MHC1. So if a natural killer cell were to come and detect MHC1 on the surface of this cell, then that MHC1 would deactivate that natural killer cell, and it would go along its merry way. So natural killer cells can kill cells that lack MHC1. MHC1 inhibits natural killer cell activity.